Hello, everyone, and welcome to today's SSR webinar. This webinar is brought to you with support from the Bill and Melinda Gates Foundation. This webinar is scheduled for 1 hour and it is being recorded. It will be posted on the SSR YouTube channel within the next week, and registrants will be notified when it is available. In just a moment, our moderator will begin with a brief introduction followed by the speaker presentation. Please feel free to submit your questions during the presentation using the Q&A text box in the bottom middle of your screen, and our moderator will read them out to the speaker when the question portion of the webinar begins. If you have any questions or concerns about this webinar, please feel free to reach out to us directly at SSR headquarters via email at membership@ssr.org or chat the webinar host. With that, I will turn things over to our moderator for today.
Welcome, everyone. It is my tremendous honor to introduce to you today's speaker, this Gates-supported webinar. Today, Dr. David Papin is joining us, and he is an associate professor in the Department of Surgery at Harvard Medical School and is an associate molecular biologist at the Massachusetts General Hospital. David's a native of Ottawa, Canada, and he completed his PhD at the University of Ottawa under the mentorship of Barbara Vanderheiden, and then did his postdoctoral training with Patricia Donahue at the MGH in Boston. Now that David is in the Department of Surgery, his research has focused on both basic biology of female reproductive development and its translational application to women's health. Dr. Papin has investigated the role of anti-Müllerian hormone in female reproduction, particularly in the context of ovarian function and disease. And his laboratory was the first to describe the contraceptive effect of supraphysiological AMH in animal models and elucidate its mechanism of action in pre-antral follicles. And we are lucky that this is the main topic of what he's going to discuss today. He is also currently developing therapeutics that are in late preclinical stage for contraception and assisted reproduction, oncofertility, and reproductive aging. All of this work has been funded by several grants, including from the NIH and from the Gates Foundation. They've resulted in numerous publications, and Dr. Papin has also been the recipient of awards, including SSR's own Narendra B. Mahesh New Investigator Award. So with that, and then finally, I also wanted to say that he, in addition to being Associate Director of the Pediatric Surgical Research Laboratories at Mass General, he is also the co-founder of Oviva Therapeutics. So with that, don't want to take up his time. He has a lot of great things to tell us. As Erica stated, please put any questions you have in the Q&A, and I'm going to turn this over to Dr. Papin. Thank you so much for joining us today.
Thank you for the kind introduction, Jennifer. I'm really happy to be here today. Hopefully, I've got everything working here. Is it okay?
Looks good. Looks Perfect.
Good. Okay. So yeah, thank you for the kind introduction. I'm excited to talk about this today, and I'm going to start with just a little anecdote. My wife also received notice of the webinar, and she told me, you made a mistake in your title. You wrote non-hormonal, and she really knows how to push my buttons. I can't believe I actually use this because it's one of my pet peeves that for some reason we often equate hormonal to steroidal, and in fact this is completely a hormonal contraceptive, anti-Müllerian hormone. It's in the name. So we really should stop referring to these this way. It's really non-steroidal contraceptive targeting the AMH, AMHR2 axis in females. Yes, so these are my disclosures. So I'm a co-founder and scientific advisor to Oviva Therapeutics, which has licensed some of the technologies I'll be talking about. So, you know what, this topic really doesn't need an introduction to the people on this seminar. I think we're all keenly aware of the need for new non-hormonal female contraceptives. But, these statistics are still worth looking at. Nearly 50% of all pregnancies are unintended, and, totaling 121 million people each year. And many of these unintended pregnancies are happening in developing regions that just don't have access to easy contraceptives. And so there's a really huge need for other forms of contraception, more options, alternatives to the current oral contraceptive pills, particularly given that, the pill is effective. It's, one of the miracles of modern science, probably on par with the invention of vaccines and antibiotics and the impact it's had on the world. But still, 1% of contraceptive users experience side effects, and for half of those side effects are more severe than they expected, and that does lead some women to discontinue the use of oral contraception, and so there's a need for alternatives. And I won't go into all the details, but this is just to remind us where we've been and where we're going. This starting with in the 50s with the discovery by Pincus et al. Of the first oral contraceptives based on steroids, which took about a decade to reach the patient, followed by other development of contraceptive methods like the copper IUD or the IUDs that release hormones. But really at this point, we're talking about 70-year-old technology, and there's been a lot of progress, incremental progress, and I don't want to minimize it because it's been important, particularly for the women that use these solutions, that dosages have changed, that the combinations of estrogens and progestins have changed, the delivery method, all these to increase both the efficacy and minimize the side effects of these contraceptives. But we're still talking about 70-year-old technology. And if we look at their utilization rates across the world, it's still relatively modest. Still most people are not on contraceptives, and a minority of them will use IUD or the contraceptive pills. And surprisingly, a very large amount will undergo surgical sterilization. It's somewhat surprising to me how much larger it is in women than in men, considering the invasiveness of the procedure. So today I want to talk about AMH and AMHR2 as new contraceptive targets and that are working on a completely different mechanism than steroidal contraceptives. And I'll also talk about new modalities, new ways to deliver contraception and their unique applications. So first I'll talk a little bit about the background of what are AMH and AMHR2 and what's the role of this hormonal axis in reproduction. And I won't go too far back, but the anti-Müllerian hormone, also known as Müllerian inhibiting substance, was first theorized by Alfred Jost. He was a surgeon in Paris, and his organism of choice, it was the giant rabbit. And he was a very skilled surgeon and would do all these very fine surgical procedures to study reproductive development. And what he'd shown is that if you castrate in utero male, you get retention of the Müllerian ducts. And the Müllerian ducts are basically what gives rise to the female reproductive ducts, the oviduct, the uterus, the cervix, and the upper third of the vagina. And then he showed conversely that if you graft a testis next to where the ovary would be in a female, you could cause regression of those Müllerian structures. And so posited that there must be a hormone produced by the testis that causes regression of the Müllerian duct. That early on, the fetus is bipotential. It contains all the structures to develop either male or female, and it is the expressed effect of the testis to cause regression of the Müllerian duct and preserve the development of the Wilson duct that gives rise to the male ductal structures. And he had access to limited technology at the time, but there was the synthesis of steroids and the ability to use them in vivo was there. And so he did put testosterone next to the Müllerian duct and showed that it was not able to cause regression. And so there had to be another hormone in the testis other than testosterone. But it took quite a while to find this hormone, and it was finally cloned in 1986, simultaneously by a group in Paris, Natalie Josso, and a group in Boston here, Patricia Donahoe and Rich Kate, who was at Biogen at the time, to finally clone this hormone and characterize it. And so although it was initially discovered as a male hormone, it was quickly figured out that it was also produced by the ovary and played a role there. And if we look at follicular development, basically the AMH hormone is produced by granulosa cell, and particularly by granulosa cells of follicles in the secondary and small antral stage. So primordial follicles don't produce it, and then the expression goes down as you approach ovulation, and it's nil in the corpus luteum. And as ELISAs were developed relatively quickly to this hormone, it was also found to be of clinical use because unlike other reproductive hormones, say steroids or gonadotropins, which cycle over time, the levels of AMH are relatively stable, but they do decrease over time. And the reason they decrease over time is that as a woman ages, she has fewer and fewer antral follicles, and those are responsible for the production of AMH. And so it's a way to measure your antral follicle reserve, your growing follicle population. And, to a certain extent, although it doesn't do a great job at this, it's also a way to measure ovarian reserve or the dormant pramidal follicles, because as you run out of them, obviously you have fewer growing follicles, and so AMH levels are down. So what is AMH? AMH is a TGF-beta ligand, a TGF-beta superfamily. There's many of these ligands. As reproductive biologists, we're definitely cognizant of a few of them, like GDF9 or inhibins. Many of those activins have demonstrated roles in reproduction. AMH, in many ways, is most comparable to the BMP family. And like most TGF-beta ligand, it's a proprotein. It needs to be cleaved to be activated, and it forms a complex. So you have a pro-domain and a mature domain. Here it is. However, unlike other TGF-beta ligand, it's very unique in that it can only dimerize with itself. So, BMPs, for example, often heterodimerize. AMH does not. It only dimerizes with itself. And also, unlike most TGF-beta ligand, the prodomain is not inhibitory. So for TGF-beta, for example, the prodomain gives it latency. So it needs to separate from it to have activity. But actually, this is the active conformation of the AMH hormone. It needs to be with its prodomain to be active. And then also, a somewhat unique property of AMH is that it signals through its own type 2 receptor. And it's the only ligand in TGF-beta family that has a 1-to-1 relationship with its receptor. So AMHR2, the type 2 receptor to AMH, only binds AMH, and AMH only binds AMHR2. So there's no crosstalk. And downstream, the signaling is basically identical to BMPs. It signals through SMAD 1, 5, and 8. So most of what we knew about AMH came from mouse models. And this publication is a particularly important one from the Temin group, Derlinger et al., 1999. And this was an AMH knockout. And the phenotype of the AMH knockout female, the male is interesting. It retains Müllerian duct, so the Müllerian duct doesn't regress. So there is a uterus in the males. But in the female, the phenotype is relatively subtle. It's only when you look at the ovary and you start counting follicles that you actually start to appreciate what is happening. And so the ovaries of mice of reproductive age, in the middle of their reproductive age, is slightly larger in knockouts. And then if you look at what happens over time, well, these mice are born with the same number of pramidal follicles. So their initial ovarian reserve is identical. But they do have more growing follicles. And you see that, especially when they are young. At the 4-month stage, you've got quite a few more non-atretic growing follicles. And then what you see over time is a depletion of the ovarian reserve that's a little bit faster. And so this is really visible when you get to late reproductive age, in the 12 to 13 month stage. Now you've got almost complete depletion. Although the effect is actually relatively subtle. So we're talking probably about single-digit percentage differences in the rate of activation. But it does suggest that it is a break, if you will, on the growth of follicles. And interestingly, these mice reproduction is completely unaffected. So that was shown relatively recently by Michael Pankhurst that, even though you have a disruption of the follicle kinetics, these mice produce just as many youngs, just as many pups, and their reproductive lifespan is identical to wild type. So they do not have accelerated reproductive aging. And that tells you a lot about what's the role of the ovarian reserve in reproduction. It's not limiting. So why did it take so long to figure out that AMH or MIS is a reproductive hormone if it was cloned in 1986? Well, I think a lot of that is due to the fact that it was a very difficult hormone to produce in vitro, any large enough quantity to do long-term, high concentration experiments in animals. So a lot of what we knew was using mouse models, transgenic mouse models. The knockout, the overexpressor it was not as useful because it expressed during fetal development and disrupted gonadal development. So there's really a paucity of research on the direct effect of AMH on the ovary, mostly using in vitro systems because it's so difficult to make. And that's in contrast to other contraceptive hormones, say sex steroids, which can be synthesized, or gonadotropins, which are short peptide. AMH is a very large protein. It's very complicated. It's a dimer. And in fact, it's essentially 2 dimers. They have a non-covalent association. And it needs to be cleaved to be activated. It's glycosylated, so you can't just make it in bacteria. So part of my initial work as a postdoc was to figure out a way out on how to make this protein, because the technology up to that really gave you very low yields and of mostly inactive protein with high levels of impurity. So this is something that I started to work on as a postdoc, and how to make large amounts of this. And we achieved this through some relatively modest changes to the protein structure, essentially optimizing the leader sequence and optimizing the cleavage site to allow high expression of a cleaved active conformation of the protein. And you can see that here. So if you have the wild type version of AMH, most of it is uncleaved and there's a lot of degradation products. If you have very little cleaved active conformation, and those that have the modifications that I've called LRMIS, you've got large amounts of cleaved protein, and then the rest was basically optimizing the chromatography to produce large amounts of it. So all throughout my talk, I peppered it with these little slides to talk about myths and some stuff that people sometimes miss about AMH, or some mean comments from reviewer 3 that I can go over now. So AMH, I just mentioned, needs to be cleaved to be activated. So not all AMH is created equal. And this is something we have to be very cognizant about when we do in vivo and in vitro experiments, is what is the AMH that we're using. And there are alternative cleavage sites. In fact, when the things that we optimized in our initial design was to reduce alternative cleavage and promote specific cleavage. And then also that AMH in the circulation exists as this conformation with its pro-domain. So this is the actual natural form of the hormone. So it's possible to purchase and use as a reagent the mature domain by itself. Unfortunately, it doesn't signal in the same way. So it can induce signaling of overexpressed receptor in an in vitro setting. But in vivo, it acts very differently. And this is not something that we discovered. This has been known since 1993. The Wilson et al. Paper is showing, for example, in the ovary, that if you use the pro-mature complex, or the natural complex of the hormone, its effect on aromatase, it's very effective in regulating aromatase. But then if you just use the mature domain, it's 100 times less effective. So you need 100 times more to have the same effect in the ovary. And this is from our own publication, although Wilson also showed this, that you actually need the pro-domain to cause regression in the Müllerian duct. So if you put this complex here with the modifications that I introduced, the LRMIS, 5 microgram per ml, the Müllerian duct completely disappears. You can see the Wolffian duct remaining behind. And if you use, for example, the preparation by R&D, which is just the mature domain at 5 microgram per ml, it doesn't cause regression. In fact, you can go 50 times higher than this, and it still will not cause regression. And so I'd like to take this opportunity, and I want to steal this thunder to just talk about this paper that just came out yesterday in bioRxiv, a study that we did with Tom Thompson, and Drew Howard is the first author. And we're finally starting to figure out why the pro-domain of AMH is so different from all the TGF-beta family members. So what we figured out is that, or what I should say Drew figured out, is that 450 million years ago, there was an insertion into the pro-domain. We don't know where it came from, if it's a piece of another gene or a virus. And it essentially gave AMH 2 pro-domains. So it has the classical TGF-beta-like pro-domain, but that's being held very far away from the mature domain, and basically as a structural function, and it keeps those ADs together, but it doesn't make contact with the growth factor part of the protein. And there's a new prodomain basically that evolved with a helical binding structure. And this prodomain not only secures the AMH, but it also keeps it active and directly makes contact with the receptor, the AMHR2, the type 2 receptor, and also ALK2 or the type 1 receptors. So it is necessary for signaling and may play a role in specifying that signaling. And so it makes it completely distinct from all other TGF-beta family members. So I encourage you to go check out this publication. The other technology that we developed that really, allowed us to do a lot of the experiments that I'll talk about is, now that we had a transgene that could be expressed by any cell and produce biologically active AMH, was to put that in a gene therapy vector, and particularly AAV9. And basically what that does is AAV9 can infect muscles, and then muscles can produce AMH at high levels, and the AMH goes into the circulation where it can reach the ovary. So it's very convenient. AMH is a secretor hormone. We don't actually need to infect the ovary or the testis to measure its effect there. We could infect any tissue, but muscles are great for this because there's very low cellular turnover, and you can get very stable expression for decades. And I asked ChatGPT to help me illustrate this concept, and it produced this monstrosity. I don't know if Mary Zielinski or any of the other editors of BOR are on the call, but if you ever need a cover picture for a special issue on Anti-Müllerian hormone, I've got the cover for you. And if not, I know a journal in Frontiers that will take it. So when we tested this gene therapy vector, we had a start finding that with about 1 month after injection and sustaining these high supraphysiological levels of AMH, we found that the ovaries had basically shrunk. They were a quarter to a tenth of the size of a normal adult, and it was immediately obvious what had happened. So, just from the knockout, we already knew AMH provided some kind of break to primordial follicle activation and early pre-antral follicle growth. And so by putting more of this break, by stepping hard on the break, we had essentially grinded follicular development in the ovary. And if you look at these ovaries, it's not that we killed the follicles. They're there. It's just that most of them are quiescent. You have a few that activate, but obviously much fewer than in the control, as you can see here. So the immediate thought that I had upon this finding, and which came just as I was starting my lab, was, how does this work, and could this be useful as a new type of female contraceptive? So I'll address some of these issues and opportunities here, and I think it's interesting to try to think of what would we want in a new contraceptive? What are the attributes of a good contraceptive? Well, in this case, we want something that works independently of steroids and gonadotropins. So, quote unquote, non-hormonal. And that, that will mean that if it works in a different way, that probably the side effect profile will be different. And I'll talk about this briefly. We want this to be safe, well tolerated at high doses, highly targeted to the ovary, no off-target effects. Again, we're quite lucky in the biology of AMH, and I'll show why that is likely the case. We want it obviously to be equally or even maybe more effective than the current oral contraceptives, which are extremely effective. And the bar that the FDA puts on new contraceptives is very, very high, higher than any other kinds of drugs. Is it protective of health? They can, the oral contraceptives have bad side effects, but they also have good ones. And we know, for example, that continued use of oral contraceptive decreases the risk of ovarian cancer. So, hopefully this could be a method of contraception that would also have some benefit to health. And then ideally, we'd have something that's more convenient than current oral contraceptive. And those are orally available, small molecule. So it's hard to make something more convenient than that, but they still require daily dosing and are quite susceptible to disruption. And so if we had something that is more favorable, easier to remember, less frequent dosage, those would be great attributes. So I'll talk first about what we think is the mechanism of contraception. There's actually multiple mechanisms at play. Then I'll talk about applications to gene therapy. And finally, can we make basically the equivalent of an oral contraceptive pill targeting this axis? So this shouldn't be news to anybody here. Basically, our current oral contraceptive work is by disrupting the feedback mechanism between the ovary and the hypothalamus pituitary, the HPG axis. So basically by replacing the normal production of estrogens, we're essentially inhibiting the ovulatory signals, the signals that lead to ovulation. And GnRH antagonists, although they're not traditionally used as contraceptive in humans, they have many different applications in reproduction. They are sometimes used as contraceptive in animals. And they basically work in similar ways, just upstream, directly inhibiting the production of gonadotropins by acting on the hypothalamus. AMH is completely different. So all these methods act on the later stage of follicle development, essentially the last 14 days of the life of a follicle, right, leading right up to ovulation. AMH acts really far upstream of this. As I mentioned, it's produced by those early growing follicles, and it acts in a feedback mechanism to inhibit both the activation and the growth of early follicles and try to reduce that pre-antral follicle population. It also is theorized to have some effects on the pituitary, some effects on dominant follicle selection, but these I'll show are unlikely to be the mechanism of contraception. So let's get back to AMH biology. Why is this a really good contraceptive potentially? It's very, very selective to a few organs, and that the reason it is because as I mentioned, AMH has only one receptor, AMHR2, and expression of this receptor is really limited to a few organs: the adrenal, the ovary, and the testis. And so, we really don't have to worry about as much about the effects of AMH on other tissues. I don't have time to go into detail on the adrenal. It's something that I've been working on for over a decade. At some point, I'll publish that encyclopedia, but I'm not particularly worried in that context. And this is in stark contrast to, say, steroid receptors. So, estrogen receptor alpha and beta, or the progesterone receptor, are expressed across a large number of tissues, and to a large extent, that's the reason why there are side effects to the oral contraceptive and why there are so many symptoms associated with menopause, with the loss of estrogens. And so this is not a problem that we have with AMH. Myth number 2. So this is something that I still have run across in papers, and for the life of me, I don't know where it comes from, but there's this perception sometimes that AMHR2 is not expressed in primordial follicles. Obviously, it's expressed there. There's mountains of evidence showing that it's expressed there, and particularly for those of us that are studying the effect of AMH on primordial follicle, if there was no receptor there, it would be very strange. And so, in every publication, almost every publication that we put out, I try to include at least one picture of showing this. And you can see it here, this in the mouse, just 2 examples of AMHR2 expression in the ovary. You can clearly see there's quite high expression in the pre-granulosa cell. This is from another publication, this with immunofluorescence, again showing expression in the pre-granulosa cell. Here are 3 different human samples showing expression in the pre-granulosa cell. So AMHR2 is expressed there. The only reason I could think of that this myth is being perpetuated is perhaps that the initial papers showing expression in the ovary used radioactive in situ, and just didn't have the accuracy to show expression in primordial follicles. So the papers say that it's expressed in growing follicles. It never says it's not expressed in dormant follicles, but I guess in the absence of evidence is taken sometimes as evidence of absence. So let's talk about the mechanism of action. I've already alluded to the fact that AMH may be involved in activation, and we know this from the knockout animals. And then when we give supraphysiological levels of AMH, there is some hints that we're reducing the rate of activation. This is using 2 different methods, either the recombinant AMH given for 40 days or AV9 given once, but looking 40 days later. And we see that the number of primordial follicles is not reduced. If anything, it's elevated, suggesting maybe there's some retention of the Herein reserve. But more importantly, you see fewer primary follicles, and presumably that's a combination of both reduced rates of activation and perhaps the increased rates of atresia. And you'll notice, importantly, that the reduction is actually relatively modest, at most 50% reduction in activation if you excluded atresia entirely. So in and of itself, it's actually not sufficient to explain the contraceptive effect of AMH. So we wanted to know what's the effect of AMH in ovaries, and I'll talk a little bit about a paper published. The first author is Marie Meinson, who was a postdoc in my lab at the time. She's now an instructor in my lab, where she looked at gene expression at the single cell level using single cell RNA-seq. And as you would expect, we can seek different transcriptional signatures in granulosa cells that are quiescent versus active versus dividing. And we see really complete different transcriptional profile when we treat with AMH. AMH has a really profound effect on transcription. And if we look at these signatures in detail, we find that as a granulosa cell, as a pre-granulosa cell becomes a granulosa cell, as it differentiates, it turns on a bunch of different genes, and we can look at it at the signature level, and we can see that even though they are technically pre-granulosa cells that are attempting to become granulosa cells in the AMH-treated group, they're not turning on those genes normally. So there's a failure to differentiate properly, even if there is activation. And if we look at the quiescent signature that we see in the control, so these are genes that are elevated in those quiescent pre-granulosa cell, we find that those same genes are even more elevated in the AMH-treated. And we've been at least talking within our lab, we refer to these as hyperquiescent. And this is an example of 2 of those genes, inhibin alpha and NR5A2, that are induced relatively early during the differentiation and activation of granulosa cell. And so you can see it in the control, basically as the activated turns on. And then you can see the failure to turn these genes on. So there's a few granulosa cells that gain the expression, but most of them do not. And that leads to very strange looking follicles. So there's an example here of a primary follicle that is starting to express inhibin alpha. In the AMH, we have a primary-like follicle in terms of oocyte size, but it's not normal. It's not turning on inhibin. And if we look at the histology, you normally go from a mesenchymal to cuboidal form. And then we've got these strange follicles here in presence of supraphysiological levels of AMH, where the oocyte is very big. In fact, it should probably be the oocyte of a primary or secondary follicle, but you still have granulosa cells that are squamous. So you've got basically a failure of granulosa cell, of pre-granulosa cell to become granulosa cell in presence of elevated levels of AMH. Another example of this, I mentioned that NR5A2 is a great marker of activated follicle, and you can see dormant primordial follicles are negative, and basically has the activated turn on NR5A2, and basically all growing follicles that contain granulosa cell are NR5A2 positive. And when you treat with supraphysiological levels of AMH, you have a failure to induce NR5A2. So you see it here and there, but for the most part, granule cells are not differentiating normally. They're not turning on the right gene. Not only that, but they're also not dividing normally. So if you look into those growing follicles, the percentage of Ki67 positive cell is granule cell is greatly reduced. If you look at the single cell level, the percentage of TOP2 positive is also greatly reduced. And if we look at the rate of growth of the follicle, so you've got oocyte size on the y-axis and follicle size on the x-axis. So usually as the oocyte grows, the follicle grows, or vice versa in the control. You can see that this relationship is disrupted by treating with AMH. So the oocyte is growing, but the granule cells are lagging behind. So myth number 3, and this is, I've put in 3 things together because they're conceptually related, but this is probably the number 1 criticism that I'll get from reviewer 3 when rejecting my grants is that as a contraceptive affecting primordial follicle, it will probably take months to take effect, and then you'll induce menopause or PCOS, which are two very different endocrine conditions. So I will argue that we do not have any data to support that. In fact, the suppression of activation, as I mentioned, is unlikely to be the main mechanism of action. The number of primary follicles is reduced only by half. What you have is more a cumulative effect of the pre-antral follicle, basically leaving you with very few antral follicle, and probably antral follicles that are not capable of ovulating. And so you don't have to wait until you deplete all growing follicles. It really has to do with the pre-antral stages. And it is rapid. So it doesn't take months, and we think that it takes months for a primordial follicle to reach the antral stage, but for the contraceptive effect to take hold, it actually doesn't take months. It takes weeks at most. And so if you got to pay attention to only one of the graphs that I'll show, pay attention to this one because it really tells us the most about AMH biology and contraception. So this is an experiment where we gave AV9 AMH to a mouse. We waited 24 to 48 hours for those AMH levels to reach hyper or supraphysiological levels in the microgram per ml, about 1,000 times higher than endogenous. And then we paired them with males, and we waited to see, how long they would continue producing litters. And so we see that they'll produce litters up to 4 weeks after being treated, and then will never produce another litter again for the rest of their lives. So it takes about 4 weeks to see the contraceptive effect. The truth is, if you calculate in the gestational period of mice, which is roughly 20 days, it really just takes a couple of weeks at most to have the contraceptive effect. But importantly, this tells us a lot about AMH biology because it also tells us that when you're pairing those males with the females, even though they have supraphysiological levels of AMH, they're still able to undergo estrus. They're still able to ovulate. Those eggs can still be fertilized. Those embryos can still implant. The gestation can still be successful. Parturition can happen. You can have healthy pups at the end of it. AMH is not affecting any of these processes in a way that would induce contraception. So it really has to do not with the follicles that are already antral and already growing and ready for ovulation. It has to do with the repletion of that pool, the next wave of follicle that will reach the antral stage. And no, you do not get menopause or menopausal-like symptoms because it's somewhat surprisingly, sex steroids are completely unaffected, particularly in cats. They're minimally reduced in mice. And you can see that here, and I'll go into further detail here, but whether you treat a mouse or a cat, and even though you have greatly reduced number of secondary and antral follicles, those follicles are still sufficient to produce normal level of steroids. And that's due in the large part to the fact that we have a really robust HPG axis that can basically maintain homeostasis and preserve the presence of estrogen, even with minimal amounts of follicles growing. And also, we do not produce polycystic ovarian syndrome. So elevated AMH is associated with PCOS. It's now one of the Rotterdam criteria for diagnosis, but it is likely not causative of the symptoms of PCOS. In fact, in our cats or in our mice, the androgen levels are normal. We do not see accumulation of cysts, which is the hallmark of polycystic ovarian syndrome. In fact, we have fewer antral follicles. And so we basically recapitulate no aspects of PCOS except maybe oligoovulation, because obviously we're using this as a contraceptive. It's preventing ovulation. Maybe that's related to the AMH effect, but elevated AMH in itself does not cause PCOS. So how does it work? So, just to summarize all this, we hypothesize that there's a contraceptive exposure window. Basically, that if a primordial follicle grows up in the presence of supraphysiological AMH, later on, it is unable to ovulate. It is still able to produce steroids, but the failure of the granule cells to differentiate normally, of the follicles to grow normally, basically predisposes them to produce follicles that are unable to ovulate, even though AMH itself doesn't have any effect on ovulation. We know that from the experiment I just described. And the effect on primordial follicle is also reversible. And we know this because we can use recombinant protein now instead of gene therapy and give it for about 40 days until we really shut down follicle development. And it takes only about 5 days to get normal levels of primary follicles. Takes only 10 days to get secondary follicles to come back. It takes about 30 days to get antral follicles to come back. And then, these mice can ovulate. We can take these eggs and fertilize them. They produce viable embryos. And so you're not actually affecting the long-term prospects of the primordial follicle, but it's really about growing up in presence of supraphysiological levels. So now I'll talk about how do we apply now this to contraception using gene therapy. And so the use case here is as a contraceptive for use in pets. And so there is this huge problem of unowned cats and dogs and overpopulation of unowned cats that is literally in the hundreds of millions across the world. So there's over 480 million unowned domestic cats worldwide. And even if you'd put every veterinarian in the world to work on this 24/7, there just is not enough of them that we could even make a dent in this overpopulation. And that's because cats are really prolific breeders and there's just so many of them. And so that's a problem not only for the cats themselves, but also for wildlife. The cats are really great predators and they put untold pressure on often endemic species, for example, in New Zealand and Australia, and they're really good at getting small reptiles and birds. They kill on the order of about 2 billion birds a year in the United States alone. And so we initially did our proof of concept of a cat contraceptive in a mouse. So even at genetic level, cats are very good at reducing mouse proliferation. So we essentially did the same thing that we had done with the human AMH and produce a transgenic version of the cat AMH. And then we tested it in nude mice so that they wouldn't have an immune reaction to the transgene, to the protein. Again, we reached microgram per ml levels in the blood. And then if we look at the ovaries of these mice, I just actually showed you the same graph earlier, we can reduce the number of growing follicles and essentially stop them from ovulating. And so the work I'll be describing has been led by Philippe Godin in my lab and Lindsay Vincent with Bill Swanson at the crew at the Cincinnati Zoo. And I'll describe one of those studies, actually two of those studies now. This one was published recently last summer. Oops, sorry. And then in this particular study, we looked at the hormonal environment of the cat to 6 months before treatment. And then we treated them with 2 different doses of AMH or a controlled dose of empty virus. Then we waited about a year and tested their ability to produce kittens. And we waited in the second year to test this again to basically ensure that we have durable contraception in the cats. And so what did we find? So we had really a vast wealth of hormonal data, unlike anything that had been done to this date. And the way we did this is, it would be obviously too burdensome and probably not ethically possible to take repeated blood samples from a cat, multiple times a week. But what we can do is take a page out of conservation biology and measure instead serum metabolites in fecal matter. And this is exactly what we did, or rather I should say the group at Crew did, literally going through thousands and thousands of samples. We had 14,000 hormonal measurements. From these, we can infer estrus phases and luteal phases by looking at peaks of steroids over background. And here's what we find. So when we this is a control animal, Michelle, and when we put her with males, she mates with the males and then she ovulates, and you can see luteal phases here. And both times when the male was introduced, she ovulated. And this is this is a treated animal, Abigail, with a high dose. You can see that when males were introduced, there was no luteal phase in neither the first nor the mating trial. And what's interesting is that, usually cats are known as induced ovulators, so they need to mate to ovulate. But often in group settings, in captivity, cats will spontaneously ovulate, and this was the case for Abigail. So before treatment, actually, she was a frequent spontaneous ovulator. She had ovulated 3 times during the observation period, and she never did so afterwards. And so if you compare now the groups, the controls, and both the low dose and the high dose, what we find is that the luteal phase frequency is diminished after treatment. So essentially, these animals are not ovulating. That's reflected in their progesterone levels as well. So during the luteal phase, phase, progesterone levels are elevated. And so what you see is a decrease of progesterone when you compare the same females before and after treatment. And this is really the proof that this works. So if we now look at litter sizes, how many pups were born from these mating studies, you can see that in the control, all the female cats became pregnant, and none of the AMH treated cats did so in either the first or second mating trial. So basically, we had complete contraception of all treated animals. And again, just to mention that, steroid levels and estradiol levels were unaffected. They're not diminished pre and post treatment in treated animals. And then we did a second study. This is not published yet, in kittens, because we're particularly in the field, you really want to intervene as early as possible, and also in veterinary clinics, you usually want to provide your contraceptive treatment, your surgical contraception, before sexual maturation. And so to replicate this, we injected the kittens 2 to 3 months of age, and they usually reach puberty at 4 to 6 months of age. So we injected them before they became sexually mature. And a similar study design, we only injected 2 controls because we didn't want to produce too many kittens in the mating trials. And then we injected 4 at the low dose and 3 at the high dose. And then very similar study design, we looked at AMH levels in the blood. They're highly elevated, microgram per ml, did mating studies. And then again, we got the same result. Both controls got pregnant, none of the treated got pregnant. But we did notice something that a little bit unusual is that actually a lot of the treated females did attempt to mate. It was different than what we had observed in the adults. In the adults, 2/3 of them were not interested in mating at all. But for whatever reason, when we treat kittens, or when we treat these kittens, a lot of them tried to mate. And if you look at those numbers, they even tried to mate more than the control. So in some ways, this is great for us because it really allows us to say that, this is not a drug that, diminishes cat libido. It really is a contraceptive. Even if they try to mate, they are unsuccessful at it. And I have some, we looked at the data, and this is hot off the press. We plotted these just yesterday, and it really shows you again that cat are induced ovulators. So if you look in the control, so this is, basically what cats will do is they'll try to mate multiple times a day for a couple of days to induce ovulation and with the intent of fertilizing and producing kittens. And then you see that here in the control. So this is basically relative to day 0 when they start mating. And usually that will induce progesterone, so luteal phase. You'll get ovulation and you'll get elevated progesterone for a while. This is what happened in treated animals. Basically, they're trying to mate, but you're never inducing luteal phase. Progesterone levels never rise. And what this tells you is that there's a failure to coitus-induced ovulation. These females are unable to ovulate. And this then translates over time into more attempts at coitus, because usually, it only takes a few days to get ovulation, and then the females will stop in the control, whereas then these kittens just keep on going because it never induces ovulation. And this, particularly in the field, if you're trying to limit animal overpopulation, is a really great mechanism because it will keep the males busy. Okay, and then finally, I want to, in the last few minutes, talk about, can we do an oral contraception for humans? And then gene therapy is great, and maybe it can be an alternative to surgical sterilization, but it's not reversible and it's not convenient. And so this is work that was funded by the Bill, originally funded by the Bill and Melinda Gates Foundation, basically that helped us produce a pipeline to screen for small molecules. And the way it works is basically a cell-based assay where we reconstitute the whole signaling axis, the type 2 receptor, we tried every type 1 receptor, every SMAD, and that induces a luciferase through a BMP response element. So it's a cell-based assay with a luciferase readout. And signaling through the AMH type 2 receptor, you get luciferase. And then we have also sort of a decoy receptor that's kinase dead, where you don't signal. And then you can see the effect here. If you have the kinase dead, you get no induction of luciferase with recombinant AMH, and you do if you have the full version of the receptor. And this was our original screen, but we've since improved upon it, and we have stable cell lines with a much greater amplitude of signal, about 20-fold. And so we did a drug repurposing library. We first screened about 5,000 compounds, and we were surprised and delighted to find 16 hits, 5 of which we validated and showed that they had activity in various ex vivo and in vivo systems. I'm showing some examples here, basically of signaling being induced. And then I want to correct some misconception about repurposed drugs. So it's not that, all these drugs that we tested have original intended targets. For example, gandotinib or ruxolitinib or JAK2/3 inhibitors. But it's not that AMH signaling is being engaged through JAK2 and 3. It's not that JAK2 and 3 is an inhibitor of AMHR2, and then you're inhibiting an inhibitor, and so you get activation. It has actually nothing to do with their intended targets. And we know this for a couple of reasons. One is that, AMH, these compounds cannot induce signaling in the kinase dead version of AMHR2. And also, we have, even as part of the library, but as part of our validation process, mostly many other inhibitors of the same target. So there's many other JAK2/3 inhibitors, and they're unable to elicit AMH signaling. So it's purely a coincidence, and it's not that surprising because by definition, these are all good for macrophages. So they're all molecules that are very good at binding kinases and modulating kinase activity. And so many of these is dose dependent. As you rise in concentration, you're not acting just on JAK2/3, for example. You're probably acting on hundreds of different kinases if you increase concentration. That's true for most drugs. So it's just purely out of luck that these would interact with a type 2 receptor, and that some of those interactions would produce agonism. And then another great proof of this is that we can give these agonists in mice that are either wild type or knockout for AMHR2. And so if you're wild type, these compounds can suppress follicle development. So you have diminished number of primary, secondary, and antral follicles. And if you put it in knockout, they have no effect. So it's not that these compounds are killing follicles or, you have to have the type 2 receptor to see the effect. So one question I often get is there then a danger of these compounds or AMH to produce Müllerian agenesis? That was the original discovery, that AMH inhibits the formation of the uterus. So what happens if a woman starts taking a contraceptive while she is pregnant with a female fetus? Well, with the AMH protein, we're quite lucky. It doesn't cross the placental barrier. We showed that initially by just giving human AMH to a mouse and showing that in the newborn pup, we could not detect this human AMH. The other thing that's protective of this is just the biology of AMH. The ovary is actually 20 times more sensitive than the Müllerian duct to AMH. So to produce contraception, we only need about 0.25 microgram per ml in a mouse and probably under 0.5 in a cat. To induce Müllerian regression, we need upwards of 5 microgram per ml. So there's already sort of a built-in safety net, apart from the fact that AMH can't cross the placental barrier. And so we actually specifically tested this. We exposed pregnant mice to doses of agonists that were known to inhibit follicle, that we had demonstrated could inhibit follicle, and also of AMH. And we showed that it doesn't cause Müllerian anomalies. So here we're showing treatment with very high doses, 3 milligram per kilogram of recombinant AMH during pregnancy. And this is the resulting uterus and the grown female pup. You see the uterus is completely normal. And roxalutinib, basically going as high of a dose as we could. Again, no effect on the uterus. So, this is obviously something to keep in mind, but I think this is something that could be dealt with pharmacologically, and there can be multiple safety nets put in place if we think this is a risk. And so we've expanded beyond this with over 17,000 compounds, basically trying to find better and better drugs that could act on the AMHR2. And I'm giving some examples here. We got really a lot of hits on the Cambridge kinase set. This is a kinase-directed diversity library, including one compound that could actually signal with a greater amplitude than the natural ligand of AMH. But these are not repurposed drugs. So a lot of these compounds would be very difficult to turn into drugs, and they really require a lot of work in medicinal chemistry to make them into drugs. And this is where, you don't have support of many foundations. Federal grants often are not supportive of this kind of drug development. In the Bill and Melinda Gates Foundation, they helped us with the pipelines, but not with drug development. So we're really, trying to scrape by and actually produce drugs and new contraceptive that could be available to women in the developing world. But we do have allies in academia, and I want to highlight again Tom Thompson, with whom we are studying the downstream signaling and the relationship between structure and signaling and function of AMH and AMHR2. And basically, we can look at nowadays, thanks to AlphaFold, we can look at differences in the folding of the kinase domain of different type 2 receptor and see how AMHR2 differs from other type 2 receptors and really try to drive at specificity. And then we can also look in silico at our agonists and see how do they actually fit in those pockets, and how can we modify them through an iterative process to make better and better drugs. And so thankfully those efforts are funded by the Massachusetts Life Science and our REPRO grant, and allowing us to keep that project alive. But really it requires a lot of effort and a lot of investment to make a drug. So in summary, we've shown that AMH and AMHR2 have been validated as contraceptive targets in multiple animal models. We've shown in mice, rats, cats, and we're working on many other species. AMH can inhibit ovulation without evidence of side effects or hormonal dysregulation in these models. And we've had cats for now over 6 years. They're nice and healthy and are quite happy. And I must say that all the cats and dogs that we've used in our studies have been adopted. Gene therapy with AMH, we've shown provides durable contraception in cats. And we've already talked to the FDA and there are hopefully clinical trials pending within the next few years. And we're still working hard at developing small molecules that are agonists of AMHR2. We have great tool compounds, but this is a little bit of a funding desert. It's very difficult. And we really need some help with this. And I've literally talked to hundreds of VCs and pharma execs. And although there's a lot of interest in women's health and, there's not a lot of risk appetite into actually making drugs for women. And sometimes I have conversations that make me wonder whether, when Pincus was working on oral contraceptive, if anybody ever told him that he should stick to rabbits and whether, the rhythm method was already good enough and didn't need to be disrupted. And then, these drugs are also have potential applications beyond contraception and oncofertility, IVF, reproductive longevity, even PCOS. So these are all things that we're exploring in the lab. So with that, I'd like to thank my team. Oh, exactly 1 hour. And there's a lot of people that are involved in this. It's a real team effort. A lot of the work that I show was done by Marie Manson and Philippe Godin in my lab. And we have a lot of former members that worked on this, a lot of collaborators like Crew, Bill Swanson, Lindsay Vincent, UMass, Guangping Gao, and Dan Wang that helped us in vector development. The Thompson Lab, we've talked about the Broad Institute where we did our screens and obviously all the funding. And I particularly want to highlight the Michelson Found Animal Foundation that have supported us from the beginning and really believed in us, even at a time where gene therapy, contraception by a gene therapy was a crazy idea. It probably would be too expensive and would never work. Well, it turns out that now it's actually cost effective and it does work. So kudos to them for believing in that from the start. Thank you.
Great. Thank you. I'm going to clap. So there's volume of clapping. That was a really wonderful talk. I want to be cognizant of everyone's time. So if you have to leave, I totally understand. Somebody else on the chat said they're clapping too. But if you have about 5 or 10 minutes, Dr. Pepin, it would be great if you could answer the few questions that have already scrolled through. So the first question is about the AMH function itself. The question is AMHR2 also expressed in corpus luteum? Yeah, so that's the first question. Is it also
neither AMH nor its receptor AMHR2 are expressed by the corpus luteum. So they're expressed by granulosa cell. We find them in theca cell as well. I didn't talk about the other cell types, ovarian surface epithelium, but the CLs don't produce AMH and they don't have AMHR2.
Okay. And then from the single cell data, can the granulosa cells from the primordial be detected? Yes, I think you're showing that.
Yes, definitely. And then that's part of the reason why we chose post-single day 6, because there's a lot of primordial follicle on a per cell basis. It really allowed us to see a lot of those.
Right. Yeah. Okay. The next question is, your cats and mice were normoandrogenic, but some studies show that AMH can reduce androgen production in human theca cells in vitro. Can you speak to what your group observed in the theca of supraphysiological model?
So totally. Like if we do these experiments in vitro, you can reduce androgen synthesis. In vivo, it does not. And again, that, I think comes back to the strength of homeostatic mechanisms between the pituitary and the gonad. We have very robust system maintaining normal steroidogenesis. And in vivo, we never see any difference in estrogens or androgens, even though in vitro, it's true that it can regulate some of these steroidogenic enzymes.
Great. Dr. Place noted that this was a great talk and beautiful work. He stated there are untoward effects of chronically unopposed estrogen exposure, i.e. in the absence of CL-sourced progesterone. How might we avoid the sequel of AMH-based contraception?
So both estrogen and progesterone levels are normal. There's only a reduction in progesterone because there's no luteal phase in the female. So the estrogen is not unopposed; it is opposed by progesterone. And then for the most part, what we're seeing is so far a protective effect of AMH. So things like I'm assuming you're alluding to the hyperplasia that may associate, particularly in animals,
right? Increased risk of endometrial cancer, those sorts of things. Yeah,
endometrial hyperplasia. So we don't see increased rates of cystic endometrial hyperplasia. If anything, we see decreased rates. So it seems to be protective, just like a gonadectomy.
Great. Since the compounds that were found in the repurposing library are JAK inhibitors and would be working on the AMH receptor at higher concentrations, is there a side effect due to their original on-target effect?
Yes, totally. And they're not all JAK inhibitors. I just chose that one as an example. There's all kinds of compounds that are affecting aurora kinases or JAK inhibitors. Yes, they're not good drugs. They're not good contraceptives because their intended targets, they're active at nanomolar levels. So they would not be good, but they're tool compounds. So they're compounds that we can modify and try to detarget, make them lose, say, their JAK2/3 activity, and modify them to retain AMHR2 agonism, and maybe even make them better AMHR2 agonists at nanomolar level. So this is what we're actually, actively working on. But again, these are difficult and expensive studies.
Yeah. Somebody noted, asked, is it true that AMH levels are high in PCOS women? And if so, does AMH contribute to the PCOS symptoms, or is it just because there are so many pre-antral follicles in PCOS women?
Yeah, I believe it's the latter. So, AMH levels are 2 to 3 times higher in PCOS. I think it's because they have a lot of antral follicles that are producing it. I don't think it's responsible for most of the symptoms of PCOS, and certainly our evidence would suggest not, because we can induce 1,000-fold higher than this, and we still don't get PCOS symptoms. Androgens are not elevated. You don't get polycystic disease. The only symptom that's recapitulated is oligoovulation. They're not ovulating, and that's great if you're trying to make a contraceptive. Right. If you're not recapitulating any of the other side effects, except contraception.
Right. Yep. An interesting question. How can we benefit from this approach and introduce it into large animal domain? Yeah.
So if we're talking about gene therapy, I think that makes it difficult because that's given on a per kilogram basis. And it's definitely something we've been thinking about. I think one way around this is actually this time, instead of targeting muscles, it'd be great to be able to target the ovary itself, and maybe locally, if you have high expression, that's sufficient for contraception, and maybe you don't have to deliver such high doses. And so this is something that I've been wanting to work on, but it's been difficult to find partners to help me with this. But I think that's would be a logical use.
There's so many questions. Okay, I'm going to ask 2 more questions, and then what I'm going to do is we can save the questions from the chat and all the comments from the chat and send those to you, and then perhaps you could feed out some of those answers. Do the large oocytes within the abnormal primary follicles that have squamous granulosa cells instead of cuboidal granulosa cells have normal transzonal projections?
Oh, that's a great question. So, I've shown these as an example of a weird follicle. They're actually relatively rare. They're just very strange. For the most part, the follicles that are growing, even though there's a few of them, the ones that are a secondary stage, the granulosa cells are cuboidal. It's just that you get these really weird primordial-like follicle. And we haven't looked at whether they have transzonal projection. That'd be a really cool thing to look at. So thank you for the suggestion.
And then I'm going to ask this last question. Sorry, I've lost my place now. What are the mechanisms for a small molecule to activate a type 1 or type 2 receptor complex? So I think they're asking about the small molecules.
So I alluded rapidly to it. We think it binds in the kinase domain. We think it affects conformation and leads it to activate a type 1 receptor. So we think there's a conformational shift and then the type 2 receptor is thought to be constitutively active. So it's really just about bringing the type 2 to the type 1. And so we think we're changing the conformation to allow this to happen.
Great. Well, I would like to say there are lots of people who did clapping on the chat. Wonderful questions. I'm sorry if I did not give your question on air, but I will make sure all of these get sent to Dr. Pepin so he can respond to people as he's able. Thank you to everyone who joined us today. And thank you to Dr. Pepin for such a wonderful talk. As I said, lots of praise for really interesting information, great data. So with that, I'm going to close this webinar and thank everyone for their attendance.